is identical everywhere. The bases which determine the sequence of the genome are where are actually not visible to the transcription factor. They are inside the double helix. The double helix backbone is everywhere the same. So how does the transcription factor know from outside what is the base sequence inside this? So this is another question. So there are many questions about the transcription factor binding to their targets, and also protein DNA interactions between in the host pathogen interactions. So these interactions are so complex, it is very difficult to predict computationally, and without actually performing lots of experiments, we hardly get much understanding of what may be happening in this. Some of the questions I tried to answer using bioinformatics in our lab. So one of them is to do a conformational characterization of DNA, that the DNA double helix is not always a ideal double helix. It undergoes certain deformations. And if you can see in this figure, that those who are not familiar with this can think of these two complementary bases in a double strand, say G and C or A and T. And from the ideal conformation, they are deformed slightly. There may be some shear, there may be some buckle, there may be some stretch. Instead of their ideal double helix, they form this kind of elastically deformed structures. And it is this elastically deformed structures are dependent on the sequence. If it is GC, the value of shear, buckle, stretch will be different. If it is AT, it will be different. If it is GG on one side, CC on another side, it will be different. And if it is GC on one side and CG on the other side, it will be different. So depending on what is the sequence at a given location, the backbone also deforms slightly, and there is what you call elastic there is an elastic deformation. And as I said, so since the basis cannot be seen by the transcription factor, what can be seen by transcription factor are these deformations which are visible at the backbone level. So we characterize this, and we developed a method called Readout, where we can take protein DNA complex structures from the protein data bank, and we can look at these structures. Okay, this is the sequence, and this is the structure that is required to bind to DNA. Then what are the sequences which can take this structure with ease, with low energy? And what are the other sequences which can take the same? So is the sequence that is placed in protein DNA complex, is the DNA sequence the very highly specific sequence, or many other sequences can form this kind of a structure? So these type of questions we analyzed, and then we created this web server, readout.cybylab.org, it should be, not ORH, and where people can upload their protein DNA complex data, and they can look at the specificity and stability of a protein DNA complex that will be formed with different sequences. They can thread large number of sequences. They can replace the DNA sequence by their own sequence and see what are the likely changes that will undergo in the protein DNA interaction energies or the specificities. So using this method, of Readout, we were able to discover a new non-canonical binding site of STAT3, and this was experimentally confirmed in Osaka University Medical Hospital, where I used to work at, in the neighborhood I used to work there. So this university, they performed the experiment based on our hypothesis, and they were able to identify and characterize this STAT3 binding site in SAA gene. So this work was published again a few years ago in BBA. And this is the whole method. Then we moved on to the next step. Then if we have a protein DNA complex, we can try to find the specificity and new non-canonical sites in the genome. But many a times we do not have a protein DNA complex. So if we do not have a protein DNA complex, can we still model what kind of conformation and dynamics that different regions of the genomes can have? So we developed this, we created a molecular dynamics trajectory for all the tetramers that are possible, and with all some flanking regions possible. So the 12 mers we simulated, and we tried to create a dictionary that if this is, for example, if this is my sequence, this is likely to be the molecular dynamics. So we created a dictionary and the software to predict the molecular dynamics directly from sequence. Instead of doing molecular dynamics in real time, we trained our model with the help of machine learning. We trained them to predict the conformational ensembles directly from sequence without performing the simulations in real time. So that's what done by training the trajectory data that we collected from the collaborators, and that worked quite well. With the help of this, we could explain some very interesting patterns of a STAT3 binding to its target. So I summarize this problem. So am I in the middle, halfway in my time? Can Professor Hassan, can you just let me try to track my time? Am I halfway?
Yeah, the time is still, it's okay.
I'm halfway, so so just to get my idea where I.
Okay, yes, you still have approximately around 15 minutes.
Okay, so I'm about halfway. Okay, so here is this problem is that STAT3 is a transcription factor, and it recognizes, as I said, the transcription factors recognize different targets, target genes in different cell types. So here it is that the data is collected for STAT3 from RNA, the ChIP-seq experiments for four different cell types. One is the macrophages, the CD4 plus T cells, T20 cells, which are cancer cell lines, and embryonic stem cells. So these four types of cells. And what is very interesting is that the number of sites that are observed in the ChIP-seq experiments, only 35 sites out of several thousand recognition sites for the STAT3, only 35 are recognized in all four cell types. On the other hand, there are large number of sites, 1,547, which are only recognized in macrophages. And on the bottom of this figure, you will see that 2,148 sites are recognized by STAT3 only in ES cells and not in other cells. So this is we wonder, we published these results in 2013 in nucleic acid research. So we wonder what makes the same transcription factor bind to different, recognize different targets and regulate different genes in different cells. And this is further insights into this. If we look at the sites individually, and if we know the canonical motif of a STAT3 is TTC and its complement, half complement GAA, with a spacer of 3 bases usually. So this canonical motif is observed in ESC cells, at embryonic stem cells, only in 28% of the sites which were observed in ChIP-seq experiment. In macrophages, this number is just 8%. In CD4 plus T cells, this number is 20%, and so on. So you would see a large number of the transcription factor binding sites do not have the canonical STAT3 motif. And then if we look at create a new motif altogether, they create different types of motifs from the sequences that are collected from ChIP-seq. So before the advent of ChIP-seq technology, this kind of observations were difficult to make. But now this ChIP-seq, 4 different independent studies produced this kind of data. So with the help of this tool that we developed, DynaSeq, and we tried to use machine learning to predict these signatures of different cell types, we were able to identify that it is not just the motif which is important, but the surrounding regions also play a role in the recognition of the transcription factor by the genome. So the target regions are here at the peak, and then information that is required in the surrounding regions dropped systematically, so much so that if we model this with the help of a regression model, we found that there are apparently 4 regions in which the 200 base pair regions surrounding the motif can be divided. In the first region, there is a information drop, and there is a different the correlation between the predicted points of this model and the observed points is 0.57. So the model shows that there are 4 modular regions surrounding the transcription factor binding modes, and then we could interpret them to be, say, the region of this is the main binding region and this is the steering region, so that the transcription factor can quickly go here, and the stabilizing region and the steering region. So 2 regions are the steering regions that help the transcription factor to tell, okay, this is not the place you have to bind, don't spend time here, go quickly to the stabilizing region. And the stabilizing region gives a stable structure for the protein, the transcription factor, to bind its target. So these results we published in 2017, and we were able to then look at not just STAT3, but in overall, but also in different cell types, they have a different profile of the shape. So in CD4 plus T, then the region just surrounding the 2 regions are important. In macrophages, all 4 regions are important. In fact, the actual motif region is less important. So probably the role of cofactors becomes predominant in the case of macrophages. In ES cells, these regions surrounding the motif are uh, roughly continuous. And same is true for cancer cells. And we already know that ES cells and cancer cells should behave more similarly to each other. And the two immune-associated cell types should behave more similar to each other. And that is what also emerged from the analysis of the DNA shape of these sequences